Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 34, where I will make a final overview of indole alkaloid biosynthesis along with metabolic engineering applications. So, in previous classes, 4 or 5 classes, I have discussed detail about the terpenoid indole alkaloid biosynthesis and metabolic engineering of the pathways leading to enhanced production of the alkaloids, and also I have discussed about the role of transcription factors, role of light in regulating the pathway, and finally, I ended up with the concept of non-natural indole alkaloid production by manipulating the pathway and feeding of non-natural substrate, and also I talked about metabolic reprogramming concept, where the normal alkaloid biosynthetic pathway along with the modified pathway both can go side by side, leading to the formation of both natural as well as non-natural indole alkaloids in Catharanthus roseus hairy roots. So, in this class basically I will show you the structures, because if you remember the previous classes I did not show any structures in order to make those concepts simple, because structures are pretty complex. So, this is an overview class. So, I will here show you the structures. Let us go to the next slide. So, basically regulation of indole alkaloid biosynthesis. So, what we have seen that the tryptophan synthase tryptophan decarboxylase, which converts tryptophan to tryptamine. So, this is TDC, and this is important enzyme for making tryptamine, which is the substrate, which is one of the substrates for strictocidin synthase, which is called STR, by joining tryptamine and secologanin. So, the tryptamine is coming from the Sikimet pathway, whereas the secologanin is coming from the terpenoid pathway originating from pyruvate and glyceraldehyde 3-phosphate, and geranyl 10-hydroxylase plays an important role because it makes 10-hydroxygeranyl, and finally, it makes loganin, and loganin converted into secologanin by secologanin synthase, and once strictocidin is formed, then strictocidin glucoside will work and it will make strictocidin aglycones, and that leads to formation of different pathways. So, one is cathinamine, which leads to the formation of ajmalicin and then of course, serpentin, whereas the other route goes towards the formation of catharanthine, and the another route goes towards the formation of tabarsonin, and then tabarsonin finally forms vindolin, and catharanthine vindolin joins and make this structure, which is the dimeric indole alkaloid vinblastine. So, these things we will see in more detail in the next slides. So, what I have also mentioned that the late step of vindolin biosynthesis, that is starting from tabarsonin till the formation of vindolin. So, this is the late step. Now, in the aerial part, that is where the leaf are situated. So, there tabarsonin is converted into vindolin by the and this requires the several enzymes 7. So, T3O, there will be T3R, so on. So, altogether 7 enzymes are involved towards the formation of vindolin, and then this vindolin and catharanthine joined by the action of peroxidase and makes anhydrovinblastine and so on. However, in the roots tabarsonin is converted into locnericin as well as horhamaricin, and then 19-O-acetyl horhamaricin. So, these and the subsequently the enzymes responsible for these pathways are also characterized like T19H, like like T19H, like MAT, these are well characterized. Another important point to tell here that the strictocidin aglycone makes the dehydrogaseocyzine. So, that is an important compound. From there the stemadenine is formed, and then from stemadenine to tabarsonin, and what I have also shown in on the one of the previous classes, that the missing step between stemadenine and tabarsonin, that I have also mentioned. Okay. Now, so this is again an overview. So, as you see here that the two enzymes which basically complete the biosynthetic steps, one is this PAS, another is the DPAS. So, that was characterized recently and the paper published in the famous journal Science in the year of 2018, and this is one, and this actually outlines the late steps, and where you see that this T3O, T3R, that those are also much later discovered enzymes, these are also there, and this is the first steps of iridoid pathway leading to the formation of secologanin, and the remaining things are also present here. One important point is that from the cathinamine, this tetrahydroalstonin and finally alstonin can also synthesized. So, this is a hypothetical view of indole alkaloid biosynthesis in Catharanthus. So, here basically the involvement of different compartments within the cells are shown. This also I discussed in detail, like there is an involvement of vacuole, there is the involvement of chloroplast, there is the involvement of cytosol and also endoplasmic reticulum, and where these reactions are carried out. So, as early as 1993, this paper published in Journal of Plant Research and by Meisner. So, this is from the Robert Vedportis group from Netherlands, Leiden. So, they have given first the overview of involvement of different compartments, and subsequently later research has confirmed that. This is the same thing, just to show you that the how complex is the structure, like streptocidin, this is streptocidin aglycone. So, and this is from different alkaloid formation like cathinamine, like azmalicin, like serpentin, vinblastine, vincristine similarly. So, this is already I have covered. So, I can leave this one, and this one also I have just told in one of the previous slides, that this is the pathway from tabarsonin to vindolin, which is operating in the aerial part or leaves, and this is the one which operates in the roots. And what I said, that the enzymes involved in the formation of horhamaricin or locnericin or acetyl horhamaricin, these have already been characterized now. Here this slide, what I would like to emphasize here, that from stemadenine one route goes towards the formation of tabarsonin, and these steps are already characterized and which contributes towards the vindolin. Other route goes towards the catharanthine, whereas from stemadenine another route originates which goes towards the formation of secodin, and ultimately it forms vindolinin. This is another indole alkaloid, of course monomeric. Similarly, from 421 dehydrocyzine gaseocyzine, acquamidine leading to the formation of perivin. These are also different indole alkaloids which also formed in different plants including Catharanthus roseus, and here it is showing the formation of alstonin and tetrahydroalstonin. So, the point here to tell that it is not only the vincristine, vinblastine, but there are so many indole alkaloids which are produced in Catharanthus roseus, and their origin from the intermediate molecules are shown here. So, this again emphasizes the complexity of the pathway. So, cellular localization of both the enzymes and intermediates, in a previous one like Meisner's paper I have already discussed, but this is 1993. So, this is a more recent drawing. So, here the involvement of different compartments like vacuole, this already I have discussed, but here interesting point is that endoplasmic reticulum is shown here, where T16 hydroxylase is located and where the reactions are carried out, followed by 16 OMT, and then the pathway moves inside the plastid, and the NMT is localized here in the thylakoid, which converts this 16 methoxy 2,3 dihydro 3 hydroxy tabarsonin into desacetoxyvindolin, which comes out into the cytosol, and then D4H converted this to diacetylvindolin, and finally DAT converted into vindolin. So, here basically involvement of chloroplast, vacuole, endoplasmic reticulum and cytosol all are important in running the pathway. This already I discussed. I have also drawn some line diagram in few of the previous classes to explain that. Azmalicin, serpentin, alstonin. So, this already I have discussed. So, I can skip it. This is alstonin, this is serpentin, which is coming from azmalicin. So, this is just a reminder that 421 dehydrogaseocycline, which formed from strictocytin aglycone, that is a branching point from where azmalin can be formed, azmalicin can be formed, which converted into serpentin later, catharanthin can be formed, and tabarsonin can be formed, which converted into vindolin, and then eventually it makes this compound. So, 421 dehydrogaseocycline is an important intermediate. Okay. This is biosynthesis of azmalin, which I also covered in one of the early classes. So, like azmalin is the final product what you find in Rauwolfia serpentina. So, which is formed from vinodin, bromelain, and ultimately it starts from 1421 dehydrogaseocycline, and ultimately it forms azmalicin through the formation of vinodin and other intermediate alkaloids. This I also covered. So, do not get daunted with the structure, just to show you that how complex are the molecules. And this is again from the structural point of view, although I have covered that, but this is a more a zoom view of the pathway, you can say that 421 dehydrogaseocycline converted into cathinamine, and which is subsequently azmalicin, and then by the action of peroxidase it converted into serpentin. This also I have discussed that stemadenin originates from 14 to 21 dehydrogaseocycline, and then one route goes to catharanthin and another route goes to tabarsonin. So, purpose is just to make an overview, so that things will percolate and settle in your mind. That is why I am showing these things again and again with the structure. And then this also I have covered that first 3,4 anhydrovinblastine is formed, then vinblastine, and then finally vincristine. Now, the difference between vincristine and vinblastine is this, that here it is CH3, which is vinblastine, whereas vincristine this is CHO. Okay, and this also I have covered. Only thing is that at this step, this is T3O and T3R, these are discovered. So, leading to the formation of vindolin from catharanthin. So, you see this is the diagram taken from 2011, that time T3O, T3R was not confirmed. So, much later diagram it is there, but intentionally I put this just to tell that with the advancement of science, new discoveries leading to more clarification of the pathway. This I have already discussed, that the elicitation signal leading to the formation of streptocytin synthase gene expression. So, where involvement of kinase, where the sometimes yeast extracts you can use. So, this is the point where elicitor binds, it could be anything, and then that activates the calcium channel, which may activate the lipase, leading to more formation of jasmonate. Then that will be received or perceived by the jasmonic acid receptor, followed by a protein kinase activity, ultimately leading to the formation of different transcription factors, including WARCA3 octadecanoid responsive Catharanthus apetellus. It could be 3 anything. So, that is the specific transcription factor for uplifting streptocytin synthase gene expression. I have discussed this with a very simplified line diagram, where basically the role of light in Vindolin biosynthesis, and particularly from the context of CRPIF1, which is a negative regulator. So, when it is under dark condition, that binds in the promoter region, and that does not allow the expression of CRGTA1 or T63H2 or DAT, which all required for the Vindolin formation. However, there is another transcription factor which maintains its basal level of activity, leading to minute formation of these alkaloids. So, the some basal activity is maintained, but then what happens that and when this happens, what happens, because Vindolin is not formed. So, tabarsonin accumulates in high amount. When light comes, light degrades the CRPIF1, Catharanthus roseus phytochrome interacting factor 1. So, it degrades, leading to activation of CRGATA, and that is a positive regulator which binds at the proper place, leading to transcription of the late genes of Vindolin formation. And what you see, that more Vindolin formation and tabarsonin is getting depleted, because that tabarsonin will be converted into Vindolin. So, this pathway is more operating. So, the content of Vindolin is more, that is basically shown by the thickness of the arrows, and here basically the thinness of the arrows indicates that less Vindolin accumulation under dark condition. So, this paper published in Plant Physiology in 2019. This is again finally I have covered. So, I can skip this. So, the different compartments involved. So, we can go. Okay, now comes to the cross section of the leaf. So, you know, at the top there is epidermis, then and then this is the epidermal layer, then there is palisade mesophyll, then there is a spongy mesophyll, these are, and then these are all. Then along with that there are spongy mesophyll, idioblast is there, and there are laticifer is there. So, the laticifer and spongy idioblast. So, these are the specialized cells which can accumulate the latex. So, and the latex contains the late steps of Vindolin alkaloids, including Vindolin and subsequently the vincristine and vinblastine. So, and this is basically okay, this is a 3-dimensional representation, this left side, whereas the right side is a cross section where it is showing that the epidermis plays important role in the synthesis of these alkaloids, including the formation of 16-methoxytabarsonin, which now moves to the palisade parenchyma, and from there it moves to palisade idioblast, which is the specialized cells where this diacetyl Vindolin to Vindolin formation takes place. Similarly, this will move to spongy mesophyll idioblast, the yellow one, where this reaction is taking place, or it can move to the specialized cells like laticifer, where this conversion takes place, because these are specialized cells having thick walls, and this actually isolate this late step of Indole biosynthetic enzymes as well as the products from the rest of the cells, because these alkaloids may be toxic to the normal cell. That is how in the evolution these specialized cells evolved. And then internal phloem parenchyma plays important role, because there the early steps of the pathways are operating. So, basically this is showing intercellular trafficking of monoterpenoid indole alkaloid pathway, and this was taken from Plant Journal 2008. Much later, review published in Phytochemistry, where the same thing it is showing, that this is the idioblast where conversion of E2F takes place. So, E2F means the late step of this Indole biosynthetic enzymes, the lactacifar. So, the yellow background cells are basically these are the either lactacifar or idioblast cells, and internal phloem parenchyma is showing with that red color, and then there is showing the upper epidermis and the lower epidermis, and the different pathways are operating are showing in the left side and right side. So, this is nothing but what I have shown in the previous slide, it is showing in worded line diagram. And it is the same thing, but what is important that if T3R is not there, then the pathway produces instead of 3-hydroxy-16-methoxy-2,3-dihydrotabersonin, it makes 3-hydroxy-2,3-dihydroxytabersonin, that leads to the formation of des-acetoxy-vindorecin. So, des-acetoxy-vindorecin again enters into the lactacifars, it converted into D-acetyl-vindorecin, and then it forms vindorecin. That is also a monoterpene indole alkaloid, quite similar to Indole structure, and this is only the new thing which is here in the slide, and this I have taken from New Phytologist. Remaining things are same. Other important point is the secretion, what I said that catharanthine is basically from the epidermal cell, this epidermis, it moves to the cuticular surface, and I mentioned because a significant amount of catharanthine is moves there, because that basically plays an important role in defense. So, that is again a relatively new information discovered, which already I have covered in the previous slides, and also I have covered that the specific transporters genes. One such transporter gene is CRNPF 2.9, which actually moves the strictocidin out from the vacuole into the cytosol, where SGD will function and it makes the strictocidin aglycone. So, that means that secologanin and tryptamine, these two enters inside the vacuole, and there lies the strictocidin synthase. That strictocidin synthase joins and makes strictocidin, and once strictocidin is formed, it should come out from the vacuole, and there is a specific transporter protein which helps to move these things out. So, what scientists they have done when they have characterized the gene, so they basically block the gene expression by using virus induced gene silencing. As a result of this, you know what happens, that the this is basically the chromatogram showing the accumulation of strictocidin. As a result of that, when 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 this gene function is disrupted, then what will happen? Strictocidin will not move out. So, strictocidin accumulation will increase. So, that is showing here in the blue one, this is the silenced line. So, because the transporter was disrupted, so the accumulation of strictocidin enhanced, whereas this is the wild type, where transporter is active. So, almost no strictocidin is detectable, because it all converted into strictocidin aglycone and subsequent downstream products. So, this was published in Nature Plants 2005, and then let us this is more or less I have covered. I have not discussed all the metabolic engineering before I end up this class. So, let me make another overview. Again I have drawn this in line diagram. What you see here, that the roots. So, root what the alkaloids accumulates in the root, actually azomycin, yombin, equivolanin. So, these compounds are accumulated in the root. So, which along with that this locnericin and these compounds. Whereas, in the leaves of the aerial part, finally, what we see the accumulation of vinblastine and vincristin. However, in the whole plant you can see all the intermediates present in the cell. So, any portion, but particularly leaf specific is these two, whereas root specific there are few which I have shown. Now, I have also spoken about the non-natural indole alkaloid production. So, non-natural means these alkaloids are not produced by the plant. So, and why they are interested? Because non-natural alkaloids may have more importance for pharmaceutical application than the natural alkaloids. Therefore, one strategy was taken by silencing the tryptophan decarboxylase. That means, tryptophan will not convert into tryptamine. So, that all the subsequent enzymes will not get the substrate, but the pathways are ready, only you block the early step. So, then what they have done, they made feeding of fluorotryptamine, and then fluorotryptamine is accepted by the root culture, when the RNAi silenced root culture, TDC silenced root culture, because TDC is not working, that is why you are putting this one and that will be subsequently taken up by the different through different pathway and leading to formation of different downstream indole alkaloids. So, that means non-natural indole alkaloids like fluorotryptamine, fluoroserpentine, fluoroazomycin, these all it is possible to make this through this technology, where you can silence the TDC and do feeding of the fluoro derivative, and that will be subsequently accepted by the normal enzymes present in the pathway and make the subsequent products which are all fluoroderivatized. Okay. And finally, I have talked about the concept of metabolic reprogramming. This is basically the metabolic reprogramming, where strictocidin synthase gene, where they made a single amino acid substitution, valine replaced with the methionine at 214 position, and then what they did. So, this is basically a mutant gene. So, mutant gene they transferred into hairy root culture. That means, they have generated co-transform hairy root culture with a mutated STR, but when but the normal pathway is still there. So, the normal STR is there along with a V214M STR is there, and then what they have done, they made feeding of chloro, methyl, bromo and fluoro already checked, and that leads to the formation of different these derivatives, either chloro or methyl or bromo of the indole alkaloids. Whereas, as the normal pathway was not interrupted, but the normal products are also there. So, in the beauty of this, you can get both the derivatized or derivatized product as well as the normal products. So, and this is the concept called metabolic reprogramming. So, where this strictocidin synthase V214, and as a result of that you can get different product formation like azomycin, tabersonin, serpentine, catharanthine. So, it basically it relaxes the substrate specificity and enabling turnover of tryptamine analogues into different product formation. So, this is a very interesting paper and this was published in Nature Chemical Biology 2009. So, and finally, the Netherlands group they also explored the possibility of producing these alkaloids in bioreactors. So, they have given a schematic diagram how the work to be undertaken. So, the fundamental study is this one which we have covered. So, where elicitation is important, they have also covered what CA3 they have covered, okay, and all these things. But then upstream processing, that is this, and then mutagenesis screening. So, this part is already done. Then important is the bioreactor design and bioprocess engineering and the subsequent downstream processing. These are also important, and if you want to produce the alkaloid in large scale. And so, the fundamental pathway is now worked out. Now, more of the bioprocess engineering needs to be, the work need to be given emphasis on the bioprocess engineering aspects and scale up, and that may lead to formation of these alkaloids at industrial level in bioreactor. So, with this I end this class. I am given you a brief overview of indole alkaloids, what I have covered, and with that I end the indole alkaloid, and we will move into the next class with another topic. Thank you.